Hello everyone, this is Dr. Divya. Topic for today's discussion is cholesterol metabolism. Cholesterol is a cyclopentopyrhydrophenanthrin consisting A, B, C rings at present. It has 27 carbon atoms and one hydroxyl group on third carbon atom, and double bond is present at between carbon 5 and 6 atom, and it also at 8 carbon it has a side chain. Biosynthesis of cholesterol. Major site for synthesis of cholesterol is liver, adrenal cortex, testes, ovaries, and intestine. 80% of synthesis takes place by liver. The enzymes involved in synthesis are located partly in cytoplasm and endoplasmic reticulum. Requirement for synthesis of cholesterol. Acetate of acetyl-CoA provides all the carbon atoms of cholesterol. For reducing equivalent by NADPH, energy from ATP. De novo synthesis of cholesterol. Primary site of cholesterol synthesis takes place in liver, and secondary sites are adrenal cortex, ovaries, and testes. And the overall reaction goes like this: 18 molecule of acetyl CoA along with 18 molecule of ATP plus 16 molecule of NADPH plus 4 molecule of oxygen converts cholesterol along with 9 molecules of carbon dioxide plus 16 molecule of NADP plus 18 molecule of ADP plus 18 molecule of inorganic phosphate. Cholesterol synthesis takes place in 5 stages. First stage called its synthesis of HMG-CoA, formation of mevalonate. Third stage is production of isoprenoid unit, and fourth stage is synthesis of squalene, and fifth is conversion of squalene to cholesterol. Step 1. Condensation. 2 molecule of acetyl-CoA condenses to form acetoacetyl-CoA. The reaction is catalyzed by an enzyme acetoacetyl-CoA synthase. Step 2. Production of HMG-CoA. 1 molecule of acetyl-CoA condenses with acetoacetyl-CoA to form beta-hydroxy beta-methylgluteryl-CoA, that is HMG-CoA, catalyzed by a reaction called HMG-CoA synthase. This HMG-CoA synthase takes place in the cytosol, leads to cholesterol synthesis. If it takes place in mitochondria, that leads to ketone body synthesis. Step 3. Regulating step. Formation of mevalonate. Reduction of HMG-CoA to mevalonate, catalyzed by an enzyme called HMG-CoA reductase. For this reduction, it requires 2 molecule of NADPH. Fourth step, formation of isoprenoid unit. Mevalonate is phosphorylated 3 times to form 3-phospho-5-pyrophosphomethylate. For this, it requires 3 molecule of ATP, and this undergoes decarboxylation to form isopentenyl pyrophosphate, a 5-carbon compound. Step 5. Synthesis of squalene, 30-carbon compound. Isopentenyl pyrophosphate isomerizes to form dimethyl allyl pyrophosphate, and 1 molecule of isopentenyl pyrophosphate, a 5-carbon compound, condenses with dimethyl allyl pyrophosphate, a 5-carbon compound, to form geranyl pyrophosphate, a 10-carbon compound, and 1 molecule of isopentenyl pyrophosphate 5 molecule compound condenses with geranyl pyrophosphate, a 10 carbon compound, to form ferrancyl pyrophosphate 15 carbon compound. 2 molecule of ferrancyl pyrophosphate 15 carbon compound condenses to form squalene, a 30 carbon compound. Step 6. Cyclization. Squalene undergoes oxidation and cyclization to form lanosterol. Lanosterol is the first formed steroid compound, and after 19 reactions, lanosterol is converted into cholesterol. Regulation of cholesterol synthesis. HMG-CoA reductase is a regulating enzyme. First kind of regulation is feedback inhibition. The end product cholesterol is in excess. It inhibits the gene which is responsible for the production of HMG-CoA reductase. And the second regulation, hormonal regulation. Here, glucagon and glucocorticoids favor the formation of inactive HMG-CoA reductase, so decreases the cholesterol synthesis. And this insulin increases. If the insulin is increases cholesterol synthesis by enhancing the formation of active HMG-CoA reductase. And the third is inhibition by drugs compared to lovastatin. It's a competitive inhibitor for HMG-CoA reductase. And degradation of cholesterol synthesis. Cholesterol is not completely degraded to carbon dioxide and water. It is converted into bile acid, which is in liver. It converts the cholesterol into bile acids, which is essential for fat digestion in intestine. And steroid hormone. Cholesterol is the precursor for steroid hormone and also precursor for the vitamin D synthesis. Significance of cholesterol. Normal level of cholesterol is 150 to 200 milligram per dl. If it is increased level, increases the risk of atherosclerosis. An important component of cell membrane which affects fluid state of membrane. Cholesterol, it is used to insulate nerve fibers. And if it is in the case of bile acid, these are derived from cholesterol, and also the steroid hormone are produced from cholesterol, and also vitamin D is also formed from cholesterol. Thank you.